Welcome back to AAT Learning Hub. Microcirculation. Microcirculation, as the name implies, is circulation at the micro level. It plays a major part in the circulatory system and comprises the arterioles, venules, and capillaries. Capillaries form the connection between the body's smallest arteries, which is arterioles, and the body's smallest veins, called the venules. It is here at the capillaries the exchange of nutrients and oxygen occurs from the blood to the tissues, and waste products are exchanged to the blood from the tissues. Now let's look at the structure of capillaries as a whole. Capillaries are extremely thin, and this allows for efficient diffusion of substances. The diameter of a capillary lumen ranges from 5 to 10 micrometers. The walls of the capillaries are composed of a single layer of epithelial cells surrounded by a layer of protein called the basement membrane. To allow exchange of substances, the capillary walls have gaps between the cells called the intercellular or interendothelial clefts. The walls of the capillaries have pericytes wrapped around them. Pericytes are cells that are essential for the regulation of blood flow and maintenance of homeostasis. So last lecture we had seen that there are three types of capillaries, which are continuous, fenestrated, and sinusoidal. Let's look at the structure of microcirculation. It has an arteriole at one end and venule at the other, and a network of capillaries in between. This is called the capillary bed. The arterioles originate by branching of arteries from the aorta. The arterioles have a continuous layer of smooth muscle cells and are innervated by the sympathetic nervous system. The arterioles further branch into smaller vessels called the meta-arteriole. True capillaries, on the other hand, branch directly from the meta-arterioles. Meta-arterioles also have smooth muscles but are not continuous and lack innervation. Venules are larger than arterioles and have weaker muscle. Despite the weak muscles, they can contract and carry blood as the pressure of blood in venules is low. Venules converge to form veins and finally end in vena cava. The capillaries at the origin point are covered by rings of smooth muscles called the precapillary sphincters. Blood flow through capillary beds is regulated by precapillary sphincters. So these can constrict or dilate to control the amount of blood entering the capillaries. This regulation is very crucial for maintaining tissue homeostasis and responding to various metabolic demands. Precapillary sphincters, they lack innervation but are controlled by changes in internal environment. Normally, the capillary sphincters are closed. When the surrounding tissues need oxygen and have excess waste products, the precapillary sphincters open, allowing blood to flow through and exchange to occur. Now, if all the precapillary sphincters in a capillary bed are closed, blood will flow from the meta-arteriole directly into a thoroughfare channel and then into the venous circulation, bypassing the capillary bed entirely. This creates what is known as a vascular shunt. Apart from this, there is an arteriovenous anastomosis that helps bypass the capillary bed and lead directly to the venous system. Now let's look at the function. Capillary hydrostatic pressure and osmotic pressure. These are the two Starling forces that control the movement of fluid in and out of the capillaries. So this is a capillary lumen and here is your tissue. So this space between the blood vessel and the tissue is the interstitium or the interstitial space. Now we all know blood pressure at the aortic level starts at 120 mm mercury. And by the time it reaches the capillaries at the arterial ends, it drops to about 30-35 millimeter mercury. The 30 millimeter mercury pressure pushes oxygen and nutrients and water out of the capillaries through the intercellular cleft into the interstitial space. The pressure that drives fluid out of the blood is called hydrostatic pressure. But larger molecules like the WBC, the RBC, platelets and proteins cannot pass through and they remain in the capillaries. The proteins in the blood, especially the albumins, are negatively charged. The negatively charged particles pull the water back into the capillaries through osmosis. Osmosis is the movement of water across semi-permeable membranes from regions of higher water concentration to regions of higher concentration of solute particles. So the inward pulling force is called the osmotic or oncotic pressure. As the blood moves forward towards the venous end, the hydrostatic pressure further drops, but the inward pulling pressure, which is the osmotic pressure, remains the same. Why? Because the proteins are still in the capillary blood. This pulls the fluid back into the capillaries. So substances which cross capillaries are water, oxygen, carbon dioxide, urea, glucose, uric acid, lactic acid, and creatinine. Now let's assume there is an infection or inflammation in the tissue. What happens? There is release of chemicals like prostaglandins, bradykinins, and these vasodilate the vessels. There is more incoming blood and hence the hydrostatic pressure increases. Also, these chemicals widen the intercellular clefts to help release WBC to the tissues to help fight infection. Now that the cleft is widened, do you think only WBCs will come out and the rest will follow a disciplined routine path? No, the proteins also get leaked. Since there is no negatively charged ions in the capillary, osmotic pressure is low now and fluid remains in the interstitium without getting pulled back. This fluid buildup leads to edema, that is swelling in the ankles or feet. Likewise, in right-sided heart failure too, since the right ventricle's pumping function is impaired, there is backflow of blood into the veins, the venules, and the capillaries. This backflow increases the hydrostatic pressure at the venous end, causing more fluid to move out into the interstitium, again causing edema. In normal individuals, however, a small amount of fluid remains in the interstitial space. Here is where another system comes into play, which draws the remaining fluid and drains it back into the venous system, thus helping prevent edema. And that super system is the lymphatic system. This is Bhumika Anand on AAT Learning Hub. Stay tuned to know more. Thank you and happy learning.